Hello, I'm Amy, and today I'm going to be giving a little bit of an introduction to flow cytometry analysis and the R package cytotypr that I have been working on. So in order to study the immune response, you have to have a method for identifying and analyzing potential immune cells. And I realized that conventional methods to analyze flow cytometry data is cumbersome and time-consuming, so I decided to develop and test a data analysis pipeline that aids in the identification of immune cell populations. So first off, what is flow cytometry? So we want to try to understand the immune cells in the body in response to, say, an infection or a vaccine. We can use a technique called flow cytometry. And the way that it works is that we have cells that express different markers such as CD3, CD4, and CD44 shown here. And these give us an idea of the function of a cell. For example, is a cell helpful in fighting off disease? And these cells can be tagged with fluorescently labeled antibodies. And this is a little bit closer to what it looks like when you have a fluorescently tagged antibodies that bind to different antigens or markers on cells. Now, when the cells are passed through a flow cytometer individually, lasers excite the fluorescently labeled antibodies and emit light that we can then measure. So for example, in this case with this cell, we would see that this cell expresses CD3, CD4, and CD44. So we can do this to look at the immune response, which can be used to diagnose disease or see if vaccines are working. So manual gating is the traditional analysis method that's used. It's typically used in a program called FlowJo. So you may start by visualizing a singular marker, in this case CD4, on our x-axis here, and then side scatter on our y-axis, which gives some information on the granularity of the cells, or how complex they are. And each little dot on this plot is a different cell. And here we can see that there are two clear populations. So we have one that's CD4 negative and one that's CD4 positive. So here it doesn't seem like it's super difficult to try to gate these cells. However, there's some other markers where there's no clear positive and negative populations. So in the case of CD28 here, one person may place a gate here showing that 18% of cells express CD28, but a different person may place it here and say that 5% of cells express CD28. The gating is just very, very subjective. But we can use controls called FMOs, or fluorescence -1, to more accurately place our gates. So when you have so many different markers and fluorescently labeled antibodies emitting light at the same time in a sample, we can make a control which contains all of the markers except for one to account for spillover. And this is so we know where to place the gate so we don't get any false positives. So in the case of the CD28 FMO, it contains all of the markers in our panel except for CD28. And because there's no CD28, we know that anything above the signal that we're showing here should be positive, because everything here is negative. So we can place this gate on our control here and then copy it onto our subsequent sample and see that about 9% of cells express CD28. So use of these FMO controls can remove some of the subjectivity and bias from our analysis. Now, when we're analyzing cells, you don't just look at a singular marker. You define cell populations by a combination of positive and negative markers. So in this case, this population 1 is a CD3 positive, CD4 positive, CD44 positive cell, which would be different from a CD3 positive, CD4 positive, CD44 negative cell. So that in this case, the difference of just that one marker can give us information on the activation levels of these cells. Now, if we have a 3-marker panel that contains CD3, CD4, and CD8, there are 2 to the third, or 8 different possibilities of potential populations. So that's every single combination of positive and negative markers. Now, current flow cytometers can analyze up to 50 parameters per cell. And in our experiments, we typically analyze somewhere around 21 markers, meaning that there are over 2 million different possible population combinations. It'd be impossible for a single person to try to explore all of these with manual gating, and some of them would be very rare or unusual populations that someone may not think to manually gate. And so in these cases, analysis can become increasingly time-intensive, can take days, weeks, and you're just never really done with the analysis because there are still so many populations you could discover. So there's clearly a need for better analysis methods that can remove some of the user bias using FMOs, are able to identify very rare populations, and that can perform analysis more efficiently. So as an alternative to manual gating, I developed a data analysis pipeline in R package to analyze flow cytometry data. And it occurs in 4 main steps: data cleaning, feature engineering, visualization, and hypothesis testing. And I'm going to go into each of these different steps in a little bit more detail. So as with all analysis, we have to start with the data cleaning step. And the data cleaning used in this pipeline is similar to what's done in manual gating, but the pipeline uses reproducible algorithms to gate the cells. So we generally start by gating on singlets. So this removes cells that may have passed 2 at a time through the flow cytometer. And so in this case, we're using a singlet gate, which removes cells where the length and width of the cell isn't close to equal. From there, we move on and gate our leukocytes. So this removes a lot of really small debris over here. So we're taking all of these cells to the right here. And then lastly, we can gate on our live cells. And so this removes dead cells that aren't of interest in our study. And we apply this data cleaning strategy to all of our samples and FMOs. So feature engineering with FMO controls is based off of the traditional FMO gating that I described earlier. But again, it uses reproducible methods for calculating where those threshold cutoffs should be. So remember, for this IL-10 FMO here, it contains all of our markers within our panel except for IL-10. So we know that any data above this threshold should be positive. So we take the 99th percent quantile of the data, meaning that 99% of the data will be on the left of this threshold and 1% will be on the right. So this accounts for a little bit of noise in the data. And we can then apply this threshold to each subsequent sample and feature engineer it. So we're converting our continuous data into binary data, which represents the positive and negative dichotomy of the cells. So any cell value above our threshold becomes a 1, and anything below it becomes a 0. So 1 is positive and 0 is a negative for the marker. We perform this thresholding on all of our FMO samples and apply the feature engineering to our samples. So after applying the feature engineering, the results show all of the different cell populations present in all of the samples. So here we're looking at 18 markers on the cell simultaneously on the x-axis, and green represents positive expression of the markers and blue represents negative expression. In this case, each row is a different cell population. So here we can see that generally we have fairly high expression of CD3, but not as high expression of CTLA4 on all of these different cells. Now, in this plot that's shown here, there were 12,122 different cell populations that were identified within a study. It would have been physically impossible to try to manually gate all of these with traditional methods. And because 12,122 populations is too many to try to analyze individually, we can filter our populations. So here we can take all of the populations and can apply a range of different filters to only look at those of interest. So in this example that I'm doing here, we filter to only cells that express CD3 and those where the cell population constituted greater than 0.5% of a sample. So this removes a lot of populations with very few cells. Now we could apply any different types of range of filters based off of the specific question of interest. So other filters could be used to look at only rare cells, for example. So we could change this filter here and say we only want to look at cells that are less than 1% of a sample. But using this filter here, it resulted in 38 different cell populations, and that's a much more manageable number to look at. And so here, this is kind of one of the plots that we can see here with the 38 different cell populations. And I am going to zoom in on one population in a minute to show kind of some of the different results of the pipeline. But first, I want to show these correlation plots. This is one of the features of the pipeline. So we can look at all of the different 38 cell populations we have on our X and Y axis and look at how they're correlated with each other. So if we see yellow, there's a high positive correlation, and purple is high negative correlation. So here we can see that populations 1 through 17 are all really highly correlated with each other. So for example, if we see as population 1 increases, we're generally seeing increases in population 2 as well. But then we can see that a lot of these other cell types are negatively correlated. So for example, as population 23 increases, population 1 generally decreases. So this can just give us a little bit of information and kind of identify different cell types that may be of interest. So here we can see that this population 21 and population 28 are not behaving like other cell types that are similar to them. So those may be ones to look into further. So if we zoom in and look at an individual population, we can see, for example, that this population 3 expresses CD3, but not CD4, not CD8, CD44, but not CD62L, and so on. And these cells can also be automatically categorized as specific cell lineages and cell types according to the marker expression. So you will have to think about the specific cell types that you expect to see in your data, but then you can code in and say, oh, I know that CD3 positive, but CD4 and CD8 negative cells are going to be double negative lineages. So you can code that in and have all the cells automatically categorized. Now, if an experiment has data from multiple time points over the course of a study, we can also explore the dynamic changes in these populations over time. So here we can see that for this population 3, at our first time point, the vaccinated group had a higher percentage of these double negative effector cells than our control group, but over time, the differences began to converge. And again, we can compare these abundances in the different cell populations over time for each of the identified populations. And in these plots, the background color for each plot gives information about the specific lineage. For example, those in blue are double negative, so CD4 negative, CD8 negative cells. Purple are CD4 positive, pink are CD8 positive, and orange are CD4 and CD8 positive cells. And so by adding these background colors, they can also help us identify some common patterns between different cell lineages. An alternative for visualizing these cell percentages, I know not all studies, you may not have different time points, and so they're using the range of different analysis methods with ggplot, and in R we can apply and look at a bunch of different visualizations for the different cell populations. So here we're showing some box plots, for example. So in many of our studies, we collect data on bacterial burden, which is a measure of the number of bacteria found in the lung. And this can give us an idea of how sick the mice are in our case. In these plots, each dot represents a different mouse at a different time point. And we can see that our vaccinated mice have about 1 log of protection, which is as expected, showing that our vaccinated group does offer some protection. Now we can take that bacterial burden data and correlate it with our immune cell data. So by looking at correlations of the bacterial burden and the immune cells, we may be able to identify immune cells that are potentially protective or non-protective. So here we can see on our x-axis, we have the percentage of cells expressing this population 3. So this is our immune cell flow cytometry data. And on our y-axis, we have the bacterial burden as log 10 CFUs. And we can see as the percentage of cells increases, generally our bacterial burden decreases. So this cell population could potentially be protective. Now this is an example of how we could correlate flow cytometry data with bacterial burden, but other numerical experimental data such as lesion scores or really any numerical data could also be used to correlate it with the flow cytometry data. And again, we can do this correlation with other experimental data for each of the 38 different cell populations. Now, when we look at the running time for cytotypr, we have a linear running time for the feature engineering algorithm. So on our x-axis, this displays the number of cells that we put into the feature engineering algorithm, and the y-axis displays the amount of time in seconds that it takes to compute the feature engineering data using the input number of cells. So in this case, we can see that with 1 million cells, it takes about 100 seconds, so less than 2 minutes, to calculate the different cell populations within the data. And we also did compare our cytotypr package to manual analysis methods. And so this is one of the examples shown here. So we gated on a variety of different populations, both with cytotypr and with manual gating. And the average difference for each of the different samples was 0.26%. So on this plot here, we have a comparison of the manual gating and the pipeline gating. And so each point represents the measurements of a single mouse for this population 3. And the point's position on the x-axis gives the population 3 measurement, based on the pipeline, while the position on the y-axis gives the population based off of the manual gating. This diagonal line provides a reference of the x equals y, where points would fall if results from the manual and pipeline gating were identical. And so we can see that they are fairly close when we compare the two together. So again, we've developed a pipeline that utilizes reproducible algorithms to clean and feature engineer the flow cytometry data, and this leads to the identification of different immune cell populations. These immune cell populations can be visualized and interrogated further in a variety of different ways. So overall, in conclusion, the pipeline incorporates controls, or FMOs, to identify positive and negative populations. It can identify very rare populations, has a fast computing time of less than 3.5 minutes, is able to correlate cell populations objectively with other experimental findings, and results are comparable to manual gating. For a little bit of additional information, we have a route map that details the workflow of the pipeline based off of the samples acquired, and shows the visualizations, hypothesis testing of interest. So here with the data cleaning, we take in our FCS files and perform our data cleaning. We then use our FMOs, and we get on our FMOs with a 99th% threshold for positive and negative populations. Another in-progress step is using, as an alternative to FMOs, if you have a clear separation of populations, like that initial plot I showed where we could see two clear populations, a negative and positive for CD4, we can use a minimum density gate to calculate the difference between the positive and negative populations. From there, we can feature engineer the data. We then do initial identification of all of the different populations. From there, we can filter to a smaller subset of populations of interest. And we can do a correlation plots to try to compare the different populations identified, and do a variety of different abundance visualizations. You can do box plots or bar plots if you don't have timeline data or time series data, or you can do time series plots if you do have multiple time points. From there, we can do various hypothesis testings, whether that's with a linear regression that I showed when we were comparing our flow cytometry data to our bacterial burden data. And there are also options to do some multiple comparison testing, applying Bonferroni, Benjamini and Hochberg, etc., to all of these different hypothesis testing methods, because we are comparing lots of different immune cell populations. You can check out the next video to look at how to use the pipeline.